cloning accumulates mutations. Long-term study reveals unexpected risks. A groundbreaking 20-year experiment has revealed that cloning, despite aiming for genetic replication, introduces a significant and cumulative mutation burden. The study demonstrates that repeated cloning leads to an exponential increase in genetic errors, eventually resulting in fatal levels of instability in cloned organisms. This discovery has critical implications for applications ranging from livestock breeding and endangered species recovery to the theoretical possibility of human cloning. The problem with perfect copies, the core issue, lies in the accumulation of mutations with each successive cloning cycle. While a single clone may appear healthy, subsequent generations exhibit a steadily increasing rate of genetic defects. Researchers found that clones harbor far more mutations than their naturally reproduced counterparts, 3 times the rate, on average, per generation. After 27 generations of cloning, large-scale chromosomal damage, including the loss of an entire X chromosome, began to manifest. By the 58th generation, cloning became unsustainable, with no offspring surviving. Why this matters? The expectation of genetic fidelity in cloning has been fundamentally challenged. The technology, once hailed for its potential to replicate desirable traits or preserve endangered species, now faces scrutiny due to its inherent instability. This raises questions about the long-term viability of cloning in any application where genetic purity is paramount. Cellular origins of mutations, the source of these mutations, is debated. One hypothesis suggests that adult cells, from which clones are derived, naturally accumulate more genetic errors than reproductive cells, sperm and egg. Another theory posits that the cloning process itself, specifically, the nuclear transfer technique, inflicts additional damage. The nuclear transfer method involves extracting the nucleus from an adult cell and inserting it into an egg cell stripped of its own genetic material. The goal is to reprogram the adult cell's DNA to initiate embryonic development. However, the physical stress of this process may contribute to genomic instability. Implications for future research. While cloning remains viable in the short term, the study underscores the need for improved techniques. Researchers suggest that gentler methods of nuclear transfer, if developed, could potentially reduce mutation rates. Alternatively, thorough screening of donor cells for existing mutations and the use of gene editing to correct harmful variants could mitigate some risks. Future applications of cloning in regenerative medicine and fertility treatments will necessitate rigorous genetic evaluation to ensure safety. The findings serve as a cautionary tale. Even a seemingly precise technology can harbor unforeseen long-term consequences. The notion of creating perfect copies through cloning is now demonstrably flawed, and future research must prioritize minimizing genetic instability to unlock the technology's full potential. In conclusion, this study reveals that cloning, while still functional in the immediate term, is not a mutation-free process. The accumulation of genetic errors with each generation poses a substantial challenge to its long-term viability, particularly in applications where genetic integrity is critical.